Assalamu alaikum. Welcome to Betlek. Today the topic of discussion is motility of small intestine. Motility in the small intestine occurs in two distinct phases. One phase is during the digestive period after food intake, and the other is during the interdigestive period when little food is present in the gut. In digestive phase, there are further two primary motility patterns. One is the propulsive and other is the non-propulsive. The non-propulsive pattern is referred to as segmentation. Segmentation results from localized contractions within the circular muscles of the small intestine. And these localized segments that are produced in the small intestine are only 3 to 4 centimeters long. So these segments contract tightly and divide the whole gut into segments of constricted and dilated lumen, and then within few seconds the constricted portions relax and then new areas constrict and form segments, and this process continues. This segmentation activity occurs to mix the gut contents with the digestive juices that are present in the small intestine, and this mixing occurs due to the back and forth movement of the gut contents within the small intestine, and this back and forth movement of the contents occurs due to segmentation. The segments that are produced at at a particular point at a given time, and after few seconds propagates to the other portion of the small intestine, and in this way segments travel over whole length of the small intestine. So this segmentation mixes the gut contents with the intestinal juices of the small intestine and causes the back and forth movement of the contents. One most important point to note here is that this segmentation motility doesn't contribute much to the net aboral propulsion of ingesta. In fact, this segmentation tends to slow down the aboral movement of materials because of the closure of the intestinal lumen in the constricted segments. So this segmentation causes the retention of ingesta and doesn't contribute much in the peristalsis or in the propulsion of the ingesta because of the closure of the materials in the constricted segments. So this was the non-propulsive movement during the digestive phase. The propulsive activity during the digestive phase consists of peristaltic contractions that migrate down in the gut in phase with the slow waves. So simply the propulsive activity is just the peristaltic activity. These peristaltic contractions are of two types. One type of peristaltic contraction occurs in the digestive phase and other peristaltic contraction occurs in interdigestive phase. So digestive phase peristaltic contractions which occur during the digestive period after the food intake pass over the shorter segments of intestine and then die out. So it causes contraction and peristaltic activity in the form of shorter segments. This peristaltic activity in the form of segments is very important because when the ingesta is pushed down the gut for a short distance, it is subjected to additional segmentation, and this segmentation decreases the peristaltic activity within that segments for short interval of time, and in this way it causes thorough mixing activity within the segments. So this was the digestive phase peristaltic contraction. The interdigestive phase peristaltic contractions of small intestinal motility are characterized by the waves of powerful peristaltic contractions, and these peristaltic contractions sweep over a large length of the small intestine, and sometimes they travel along the entire length of the organ. While in case of digestive phase peristaltic contractions, we have seen that the peristaltic waves pass over the small intestine in the form of short segments. But it is not a case in the case of interdigestive phase of small intestinal motility. Instead, the powerful peristaltic contractions sweep over the entire length of the organ in case of interdigestive phase of small intestinal motility. So these waves that traverse the entire organ are called migrating motility complex or migrating myoelectric complex. This migrating myoelectric complex begins in the duodenum as a group of slow waves, and this group of slow waves stimulates intense action potential and muscular contraction activity. As a result of this intense muscular contraction activity, there occurs strong waves of peristalsis, and these strong waves of peristalsis migrate down the intestine. Some of the migrating myoelectric complex die out before reaching the ileum, but some travel the entire length of small intestine. At the end of the small intestine, there is an ileolic sphincter at the junction of small intestine and large intestine, and this prevents the reverse movement of colon contents into the ileum. This ileolic sphincter consists of well developed ring of circular muscles, and it remains constricted at most times. In addition to this muscular ileolic sphincter, in many species there is a flap of mucosa, and this flap of mucosa acts as one-way valve, and in this way it further blocks the movement of colon contents back into the ileum. When the wave of peristalsis reaches the ileum, the ileolic sphincter relaxes, and this allows the movement of materials from ileum into the colon. But when the pressure in the colon increases, then the ileolic sphincter constricts more tightly, so that the contents of colon so the contents of the colon may not be reversed into the ileum. Now let's move to the motility in colon. The colon has multiple functions which include absorption of water, electrolytes, and storage of feces. And third function, which is present only in some small ruminants and non-ruminants, is the fermentation of organic matter that escapes digestion and absorption in the small intestine. So fermentation of organic matter occurs only in horse and rabbit, while in case of cattle, they are foregut fermenters. So rabbit and horse are hindgut fermenters. In case of canines, there is no need of fermentation products, and they have simple colons. As horse and rabbit are hindgut fermenters, so they make extensive use of fermentation products for their nutritional needs and have large and complex colons for this purpose. On the other hand, dogs and cats don't rely on the fermentation products and therefore have relatively simple colons which perform the function of absorption of water and electrolytes, and helps in the storage of feces. Colons of all species undergo mixing activity, and its importance is that because the mixing and circulating are very important to both absorptive and fermentive functions. So both the absorption and fermentation in the colon require the mixing and circulation of the materials present in the colon, and therefore the colons of all species undergo mixing activity. This mixing activity is achieved by the segmentation contractions along with other types of motility. And in many species like horse, colonic segmentation is pronounced, and in some areas it results in the formation of sacculations known as hostra, which are visible even after death. These hostra in the colon of horse give segmented appearance, and the hostral contraction is activated by the presence of chyme, and it serves to move the food slowly to the next hostra, and along with mixing the chyme to help with the water absorption. So the hostra of the colon of horse cause the movement of food to the next hostra, and also cause the mixing of the chyme, and in this way it helps in the reabsorption of water. There is a particular characteristic of the motility in colon, and that particular character is the retropulsion or antiperistalsis. This type of peristaltic contraction migrates towards the mouth, or we can say that this peristaltic contraction migrates orally. The opposite of the normal This is opposite of the normal peristaltic movement, and such motility results from colonic slow wave activity. These antiperistaltic contractions occur in the form of segments, and in these segments slow waves migrate in oral direction. As these antiperistaltic contractions move aborally, They force the material to be accumulated in the proximal portions of colon, and in this way antiperistaltic contractions hinder or impede the movement of ingesta so that it can be mixed intensively and properly. I have said that antiperistaltic contractions occur in segment and in which slow waves migrate in oral direction. So the origin of these slow waves in the colon is similar to the gastrointestinal tract, and that origin is interstitial cells of Kajal. But the colonic enteric nervous system has one exception, and that is that this enteric nervous system can influence the interstitial cells of Kajal in such a manner that it can shift the site of origin of slow waves and direction of their propagation. Due to this exception, the slow waves of antiperistaltic contraction move orally instead of aboral movement. So under the resting conditions in colon, these slow waves originate from pacemakers or interstitial cells of Kajal. In the case of colon, these pacemakers are not the anatomical structures, rather they are areas defined by the activities of enteric nervous system. Because the colonic enteric nervous system has exception that it can shift the site of origin of slow waves. Therefore, these pacemakers are not always the same areas. They can disappear and form in different locations in response to the need of different motility patterns. So the antiperistaltic contraction appears to be stronger near the pacemaker, and the pacemakers therefore represent the sites of high resistance to the flow of colonic ingesta and help to inhibit the movement of ingesta. Because of the continued inflow of the material from the ileum into the colon, some ingesta can escape the retropulsive or antiperistaltic motility, and it can go into the areas of propulsive motility or peristaltic activity, and in this way it can proceed along the colon. In addition to this, there are certain periods of intense propulsive activity, and these propulsive activities or peristaltic waves involve the entire colon, and these are called mass movements, and in this way they frequently involve the distal translocation of entire colonic content. So as these intense waves of propulsive activity travel along the entire length of colon, they cause the distal translocation of entire colonic contents. In case of dogs and cats, the colon is a relatively simple organ as compared with horse and rabbit, and it consists of a short cecum a short cecum which consists of an ascending part, transverse part, and descending part. During the resting phase, there is a colonic pacemaker at about the junction of transverse and descending colon. This pacemaker gives rise to antiperistaltic activity in the proximal colon with resultant accumulation of ingesta in the cecum and ascending colon areas. The material which enters the carnivorous colon is of fluid consistency. Therefore, it is thoroughly mixed in the ascending and transverse colons, and much of its water and electrolyte content is absorbed by the ascending and transverse colon. And by the time it reaches the descending colon, it becomes semi-solid feces. So the fluid consistency of the carnivorous ingesta becomes semi-solid feces, or is changed into semi-solid feces when it reaches the descending colon. In the descending colon, there is moderate peristaltic activity, whereas the distal colon and rectum are usually empty and constricted.